lots of innovations and technology development that we've heard about today, which are really changing the way that we're managing our horses. And I really believe now with all of these technologies and with the technology that I'm going to talk about, which is genomics, that we are really living in a new era of breeding. For hundreds of years, breeders have relied on evaluating pedigrees to best estimate or best guess the genes that have been passed down from ancestors in that pedigree to the individual to determine the likelihood of that individual being a champion. What we can now do, alongside that, as well as the pedigrees, that's also been coupled with evaluation of the conformation of the horse. But it's important to recognise that for as long as thoroughbreds have been bred, genetics has been recognised as being an important part of the decision-making process and understanding of the individual. A lot has changed in the last 10 years. 10 years ago, the horse genome was sequenced, and these are images actually of the lab where the genome was being deciphered for the horse. This has allowed us as genomic scientists or equine geneticists, has given us the technologies, the platforms that are on a par with human medicine in understanding genes that are contributing to traits of importance in humans, in particular health traits and traits relating to medicine. And for the horse, we're interested not only in health and disease traits, but also traits that would be of economic value or economic relevance. So we now have the tools that enable us to study the DNA of the horse and understand how differences in the DNA can have an effect on inherited traits. We now know the 3 billion letters of the genetic code for the horse. There are 3 billion of these units, the GATC units, which are the units of the DNA. And in the thoroughbred, there are about 1 million of these letters that actually change. And what we are trying to do is to understand where there's a change in the DNA code, how does that affect the physical phenotype of an individual. So it may look a little bit like trying to find a needle in a haystack. It is to a certain extent, except that we can use what we would, I suppose, the equivalent of a metal detector. Using certain tools and certain techniques, we can home in and zoom in on these certain regions of the genome and identify then the genes that are there and the DNA differences that are underlying traits that those genes are contributing to. So while this sort of approach has been taken in a range of species and then breeds within species, the thoroughbred in particular is an extremely useful model system to understand genes contributing to traits because of its quite unique genetic structure. The thoroughbred comes from a very small number of founders. On the paternal lineage, the Y chromosome, just 95% of paternal lineages traced to just a single stallion, and 72% of maternal lineages traced to about 10 mares. So coupled with this very small foundation gene pool, the thoroughbred, as we know, has been a closed stud book. And so there hasn't been gene flow in and out of the breed for about 200 years. As well as this, there's been very intense selection for traits of importance that have been selected at each generation. And at each generation, that selection means that those genes are propagated with individuals that are selected to go on and remain in the breeding herd. So all of these things together allow us to much easier, I suppose, have a stronger metal detector to find the differences that in the DNA code that are having an effect on traits of interest. I started research in 2004. This was even before the horse genome was sequenced, to understand genes contributing to race performance traits in the thoroughbred. And it was during this research that we made the discovery that we now refer to as the speed gene, which is that we could read the letters of the DNA code of a particular gene and make a prediction about how that horse is likely to perform on the racecourse. The gene, in fact, is the myostatin gene. And it is the same gene that causes unusual muscling in a range of mammalian species, including cattle, dogs, mice, sheep, pigs, even in humans. So this bull on the left that's a Belgian blue bull, and he has what's known as the double muscling trait. He looks like that because he has 2 copies of a mutation in his myostatin gene, one inherited from his mother and one inherited from his father. So he's what's known as homozygous, or double copy for the myostatin mutation. This dog in the middle, believe it or not, is a whippet. He's known as a bully whippet, and he looks like that because he has 2 copies of a mutation in his myostatin gene, one inherited from his mother and one from his father. Now that's actually detrimental to racing performance for these dogs because they have too much muscle bulk. But dogs that have 1 copy of the mutation, or what we refer to as heterozygotes, are better racing dogs than dogs that have no copy of the mutation. This mouse here is known as the mighty mouse. He actually has no myostatin at all. It's been genetically removed from his genome. And myostatin acts as a negative regulator of muscle development. So it acts almost as a brake. You can imagine it as a brake. And when it's not there, the brake is released and allows this unusual muscle growth. When I started this research, we knew in these species and in a number of others about these mutations. And I asked the question then, well, is there any evidence for mutations in the myostatin gene in horses? And when I searched the literature and I looked in online databases, there was no evidence at that point. So what we did is we read the sequence of the letters of the DNA code for the myostatin gene, which is about 8,000 units of information long. And when we did this in a set of thoroughbreds, we found that at a particular position in the myostatin gene, this is a schematic of a chromosome, but in a particular position of the myostatin gene, the letter could either be a C or it could be the letter T. And we refer to these as alleles. So if an individual inherits a C-type chromosome from the sire and a C-type chromosome from the dam, then we refer to their genotype or their genetic type as CC. If they inherit a C from the sire and a T from the dam, or the other way around, then they're CT. And if they get a T-type chromosome from both the sire and the dam, then they're what we refer to as TT. Now, every single horse must be one of these 3 types, either a CC, a CT, or TT. Not just a thoroughbred, but all horses must be one of these 3 types. So we asked the question then, is there any difference when we look at different subpopulations within the thoroughbred? Is there any difference in the frequencies of these types when we look, for instance, at elite performers versus poor performers, or when we look at sprinters versus horses that are more suited to stamina or endurance type racing? And I'm talking about within the thoroughbred population, we know that there is variation in distance aptitude. Horses that excel over 5 furlong races are quite different from those horses that excel over 1.5 mile or 2 mile distance races. And what we found was that there was a very clear and very strong statistically significant association between the genetic type or genotype and the best racing distance for an individual horse. The CCs were best suited to the short distance races, the CTs to the middle distance races, and the TTs to the long distance races. So we referred to this then, these differences in the myostatin gene as the speed gene. So we can use this information now to predict the likelihood of a horse performing optimally over certain distances on the racecourse. These are the distances for a set of group winners in North America. So the CCs have the shorter distance range, the CTs the middle distance range, and the TTs the longer distance range. They're quite different distance ranges in different regions of the world because of the different race patterns in each region. And so therefore, we have to recognize, we do recognize that it's not just genetics that's contributing to distance, it's the opportunity to race over certain distances as well. So the environment does play a minor role as well in this particular situation. Oops. One consideration was that when we first did the study, we looked at a single gene. This is what we refer to as a candidate gene study, where you look at a single gene at a time and you hypothesize that that gene is going to have an effect on the trait of interest. But if you take an agnostic approach and you don't expect that there's any particular gene having an effect on the trait, and you look across the entire genome, this is what we refer to as a Manhattan plot, and each of these dots represents one of these changes, a C to T change in the genome. And what we see here is a very singular peak on chromosome 18. And when we take our molecular magnifying glass and look at where those particular changes are located, they're all located in the region of the myostatin gene. When we ask the question, what's the difference between horses that are best suited to sprint racing and horses that are best suited to stamina distance racing? So it's very clear that what we now refer to as the speed gene or the myostatin gene has a major, almost singular influence on distance aptitude in thoroughbreds. There are some other genes that are having modifying effects, but unusually and remarkably, it seems that there is a single gene. This is not something that we were expecting. We would expect that there are multiple genes that are contributing to performance traits, and in fact there are multiple genes, in fact probably thousands, even tens of thousands of genes that are contributing to the overall anatomical, physiological, and metabolic adaptations that enable elite racing performance. But for this specific trait of distance, this gene is having a singular effect. The reason for that is that not only is it responsible for muscle hypertrophy, but studies have shown that it's responsible in the thoroughbred for the laying down of the different fiber types. The CC horses that are the sprint type horses have a significantly higher proportion of type 2B muscle fibers, and these are the fast glycolytic muscle fibers that are required for high intensity short bursts of speed. Not only that, is we looked at a set of, this is almost 200 yearlings in a single preparation and early training facility in Japan, and we measured their height to weight ratio as a proxy indicator of muscle mass. We found that the growth trajectory for the CCs was significantly greater than for the TTs, particularly when these horses first saw or first were exposed to an exercise stimulus. So what happens when exercise stimulates the muscle, the brake is on, and as the muscle reacts to exercise, the brake comes off, and therefore muscle grows. And this seems to be a stronger effect in the CCs than in the TTs in the early stages of training. As well as that, we've recently shown that CC horses have greater activity of a particular mitochondrial enzyme than TT horses. And this may have an effect, or seems to have an effect, on the efficiency of the electron transport chain in the mitochondria, which is responsible for converting oxygen to energy that can be used during exercise. The consequences of all this is that CC horses tend to be more early maturing, precocious horses. And this is really where the value of this information is used in the industry now, in having this information. So what we found is that CC horses have their first start on average 80 days or 2.5 months earlier than TT horses. And what was interesting is that CC horses, while CC horses had their first start 2.5 months earlier than TTs. They had their best race on average 5 months earlier than TTs. 1 in 4 of these foals will be a CC, half of them will be CTs, and a quarter of them will be TTs. Now remember, all of these foals will have identical pedigree pages because they're out of the same mare by the same stallion, but genetically they can be very different. And this means that they will mature and grow as young horses quite differently, and they will perform very differently on the racetrack. We, as Plus Vital, have clients in all of the major racing regions of the world, and people are using this genetics to inform breeding, training and racing decisions on a daily basis. Many people elect to keep the information, use the information for themselves, but this was a very public discussion or debate about the use of this genetic information when the connections of Galileo Gold last year, who had won the Guineas, decided to use our genetic test to assist in their decision making to determine or to decide whether Galileo Gold would compete in the Derby, which is another half a mile distance. And because he was a CC and we had only tested a very, very small proportion of the population of CCs that had won over distances greater than a mile, in the order of 1 to 2%, the decision, along with the trainer and the owner's views, was to not compete this horse in the Derby. We have a number of examples of how this information has improved the bottom line, or the top line, I should say, actually, in terms of operations. We had an owner breeder who decided that if he produced fewer TTs, then he would, on average, increase his opportunity for racing horses as 2-year-olds because of the restriction of race distances for 2-year-olds and the precocity of CCs versus TTs. So he reduced his proportion of TTs from 16% to less than 5%, with the knowledge of the genetic type of all of his mares and the stallions that he was using, and in the space of 2 years trebled his 2-year-old earnings, strike rates, and his earnings per run by reducing the proportion of TTs using this genetic information. Now, over time, this happens and has happened naturally. This because of the pattern of racing, which I mentioned earlier on. In the European population, about 25% of the population are CCs, half are CTs, and 25% are TTs. And that's because we have a wide-ranging race pattern system here, where there are high-value races in the middle to longer distance range. Whereas in Australia, there's significant emphasis on early 2-year-old speed and precocity, and therefore over time, or in the last 100 or so years that horses have been racing in Australia, there's a significantly higher portion of CCs and CTs in that population. So over time, this will happen naturally, according to the natural laws, the laws of selection in the population. But what we can do is using this information, or what breeders can do using this information, is to accelerate this rate of change within their population to more consistently produce the types of horses that they desire. The speed gene, or myostatin, is probably the best studied gene relating to performance or any health trait in the thoroughbred to date, in terms of publications, not only from my research group in UCD, the company Plus Vital, but also other academic research institutions internationally. But of course, it's not the only gene. It is specifically influences muscle type and muscle growth, and therefore the distance phenotype. But there are many, many other genes. Of the 25,000 genes in the genome, there are many of those genes that will significantly influence many other traits that are of economic importance. And not just elite performance, but many other traits in terms of behavioral traits, nutritional requirements, trainability, response to stressors, as well as health traits. And a number of these were mentioned earlier on, like laryngeal neuropathy, bleeding. We haven't mentioned tying up or OCDs. All of these have significant heritable components that have been long recognized by breeders and by individuals who manage horses. And in some cases, there are studies that have shown through pedigree analysis the amount of variation in the trait that is attributed to genetics. We refer to that as the heritability. So we know all of these have underlying genetic contributions. The question is, can we unravel all of this information and package it in a way that we can make predictions about the risk or the chance of developing or having any of these traits? We can now assess in a single go in a single time from a single DNA sample 670,000 of these genetic markers at a time. Using our research sample set of over 8,000 horses, we've tested 15,000 horses, and we're waiting for those horses to mature so that we can generate the phenotypes or the race phenotypes for them. And employing highly sophisticated machine learning predictive modeling algorithms, we can now package this information and use this information to make predictions about how horses will perform on the racetrack. I'm just going to give you one example, and this is a test that we've developed for the chance of a horse having a race course start as a 2-year-old or a 3-year-old. Somewhere in the order of 30 to 40% of thoroughbred foals born don't ever make a race course start. And we've identified sets of genes that allow us to predict whether the chances of a horse having a race course start. When we determine them as high potential for a start, high genetic potential to have a race course start, medium and low genetic potential for a race course start, we find that horses with high potential were 2.6 times more likely to have a race course start than low potential horses. But in the sales ring, buyers were paying just as much for the low potential horses as they were for the high potential horses. Genetics, importantly, is not a silver bullet. It is a part of the package. You have to start with the raw material. The genetics is the raw material. And how you then manage that raw material really is the key. And that's where genetics plays a role alongside horsemanship and the management of the horse. And this is absolutely critical. The idea is that you can use this genetic information to maximize the environment for that horse, to get the best out of that horse and to maximize its genetic potential. Many of these, there are researchers internationally working on some of these traits. Some of these traits we are working on in our research group, both in the university in UCD, but also in Plus Vital. And in the next very short number of years, there will be predictive genetic tests for risk or chance of these traits. The technologies are rapidly advancing, but this is not the rate limiting factor anymore. We have the tools where we can advance and understand the underlying genetics to these traits. Really, the rate limiting step to any of these is really the cooperation with the industry in terms of diagnosing or phenotyping horses for these traits. Strongly believe that the use of genomic prediction tools will become standard practice within the industry in the next 5 to 10 years. There is considerable resistance at some levels in changing how things are done, but at other levels we have a thriving business where we have many people that are using this information extremely successfully to their advantage. I'll leave it to the great Charles Darwin to have the last say. He wrote in his epic Annals of Animals and Plants Under Domestication that although man doesn't cause variability and cannot even prevent it, He can select, preserve and accumulate the variations given to him in any way he chooses and can certainly produce a great result. So using genetics, really what we're trying to do is to identify the natural genetic variation that is there to assist in our choices. Thank you very much.